हेलो सो वेलकम टू बाय लाइब्रेरी गाइस। जितने क्वेश्चंस हमारे बच गए थे विल डू देम। सो टिल 50 क्वेश्चन वी हैड ऑलरेडी डन एंड नाउ आई विल बी डीलिंग विद क्वेश्चन नंबर 51 टू like question number 72 because I don't have much time, so I want to be constant, therefore I'm doing this. So the first question is protein sequence comparison is more sensitive than nucleic acid sequence comparison because see the thing is whenever you have a protein, because protein has to carry out all the essential functions, then also because of this reason also protein sequence comparison is more essential. But they are asking why is it more sensitive than nucleic acid sequence comparison. So it is because of the codon bias. So what is codon bias? The preferring one codon over the other. So why codon bias is important as well as sensitive? Because it has a very essential role in evolution, because by this you can know that in certain species, say for example there are two species, so you can compare that where in evolution some of the codons were preferred, that is some of the those proteins become we call them conserved proteins. So it becomes more sensitive because of the codon biasness. Whatever is preferred by the system or preferred by the cell is advantageous for the organism. So moving to 52nd question, that is sickle cell anemia is an example of SNP. SNP is single nucleotide polymorphism, that is difference at the level of a single nucleotide of the options are A to T mutation, T to A, G to C, or C to G. So they are asking at the level of DNA. So it is A to T mutation because it becomes from GAG it becomes G to G. You can also watch one of my videos where I have explained, I think in 5 minutes, about this whether it is transition or transversion. So 53rd question is which of the statement about translation is false. So we all know about translation. It is the basic process by which protein is synthesized inside a cell, and we can't say that translation leads to the synthesis of a functional protein, because for a protein to become functional, splicing as well as post translational modifications are required. So they're asking about the false statement. So the first statement says during translocation in the hybrid sites model, the tRNA attached to the nascent polypeptide chain is in the P site of the small subunit and the A site of the large subunit. So this peptidal transferase or the peptidal tRNA, this peptidal tRNA binds to P site. So it's not like that some of the tRNAs are attached to the a small subunit, or some of them are attached to the large subunit, right? So it is one subunit as like integrated subunit. So the first option will be false, and rest you all can see all the statements are right. Saying B is saying fusidic acid prevents release of EFG-GTP, yes it does so. And C is puromycin, from puromycin PP, that is puromycin premature release of polypeptide chain. And D, IF3 preferentially bind to 30th ribosome, it is also correct. So false will be A. Now we come to 54th. 54th is saying for the folding of linear polypeptide into a compact tertiary structure, that is tertiary means what? Tertiary or functional structure, globular in nature, the change in entropy is known to be negative. So it is saying that for a protein to be functional, the change in entropy will be negative. Also the question is asking, in order for the folding process to be thermodynamically feasible, the overall change in enthalpy based in intermolecular interaction should be. So for the entire like the process to happen, the formula is delta G is equal to delta H minus T delta S. So as far as I remember, the formula is delta G is equal to, you guys can also remind me, delta H minus T delta S. So if this is, it is saying that this is already negative, right? If this one will also be negative, then only the entire delta G will become negative. So it is, the answer will be negative. Now comes 55 question. If the equilibrium constant for a chemical reaction at 20 degrees centigrade is 20, then standard free energy change associated with the reaction will be. So the formula for this calculation of standard free energy change and its dependence on a temperature is like delta G naught, free energy change is represented by G naught, is equal to minus RT ln KEQ. So the things you have to keep in mind is, all of you will be able to solve the question when this question comes, but the thing you have to keep in mind is the units. So the answer is in kilocalories, but you have the value of Rydberg's gas constant in kilojoule. So you will have to search and you write in your copy what is the value of R in kilocalories. So it is 1.98. So R in kilocalories will be 1.98. So if you will multiply and put it in the equation RT-ln minus RT-ln K EQ, then the answer will come as minus 1.74 kilocalories. You can also follow my Telegram channel. There I will post the solution of this question. Okay, rest I'm telling you the formula. If you want to solve, you yourself can solve this question. Then comes question number 56, which is saying CAMP, that is cyclic adenosine monophosphate, regulates the lac operon by. We all know that wherever CAMP is involved, or wherever CAMP is involved, then it will also involve one of the protein called as CAMP activating protein, right? So catabolite activator protein. So it will bind with catabolite activator protein, it will bind with cap and form a complex such that it enhances the transcription. So this binding of CAMP to CAMP is responsible for positive regulation. So let's look for the option. A is binding to the operator to turn on the transcription. No, it doesn't directly bind to the operator. B is binding to the lac repressor to prevent transcription. No, it does not prevent transcription. So the C option is combining with the catabolite activator protein cap to form a complex that enhances transcription upon binding to the promoter. So C will be correct. Now comes 57th question, that is co-transport of nutrients across the intestinal cell membrane is an active process that can move glucose against a concentration gradient. So it's an active process, right, in intestine. Whenever this sodium transport takes place, or the co-transport of nutrient, that whatever we eat, it gets transported as a it gets transported with the help of energy. That is why it is called an active process. So the energy requiring step for co-transport involves, the A option says, the sodium potassium ATPase that pumps sodium ion from the cell into the lumen of the intestine. No, this sodium potassium ATPase does not pump sodium. It pumps out from the cell, but does not pump into the lumen of the intestine. So we all know about the sodium potassium ATPase pump. Does what? 3 sodium ion outside the cell and 2 potassium ion inside the cell. So wherever the option will be like excluding the sodium ion from the cell, that option will be correct. So B is the permease that allows glucose and sodium ion into the cell, again incorrect. Then C is the permease that pumps glucose from the cell into the blood, again incorrect. So then the thing is so the sodium the option that was incorrect here is permease, because outside the cell there will be blood. So pumping out sodium eventually means pumping out sodium in the blood. So the fourth option is the sodium potassium ATPase that pumps sodium ion from the cell into the blood, maintaining low sodium ion levels in the blood. So what does it do? It pumps sodium ion outside the cell into the blood, and therefore low sodium ion is maintained in the cell. And you also remember the fact that glucose and sodium ion transport takes place in a co-transport manner. So whenever a patient is hospitalized, then along with glucose, that patient is also kept on sodium, such that this co-transport can be maintained. So that is the reason they are also given sodium along with the glucose. So D option will be correct here. And 58th question says, then the question 58 says, the endogenous GTPase activity of G protein serves to, we all know that whenever a protein is GTPase means GTP cleaving. So it will hydrolyze GTP, returning the G protein to a pre-stimulated level of activity. So the option number D will be correct. See, in some questions, when your theory is very strong, then you don't have to look for other options, right? Your eyes, they naturally go to the correct option. Like this question, endogenous GTPase activity of G proteins. We always, whenever we start studying signal transduction, first step we read is about GTPase, GAP proteins, right? So the 59th question is talking about cytochelazins. These are drugs that interfere with actin polymerization into microfilaments. If you don't know this information, kindly write it down, that cytochelazins, they inhibit the actin polymerization into microfilaments. If you add cytochelazin to cultured mammalian cells that have just begun mitosis, so what is most likely to happen? So if you have cultured your cells and your cells are beginning their mitosis, and but just before this mitosis process, like just with the mitosis process, you have added cytochelazin. So what will happen? First option is the cells will arrest at mitotic metaphase. It's the thing is, if it interferes polymerization into microfilaments, it will not arrest your phases, right? It will arrest the phase where your cells get separated. So it will only act at the level of cytokinesis. So the option will be the cells will complete mitosis. There will be no harm to the cell and arrest at cytokinesis. So whenever your cell will be separating, like here a prophase band is present, prophase band is getting formed, there are a lot of microtubules here, and this cytochalasin does what? It inhibits this polymerization. If it will inhibit this polymerization process, then your cells will not be able to separate, so it will arrest cytokinesis, right? So the option number C will be correct. Then comes 60th question. Which of the following is not a part of the method used in single locus probe analysis of VNTR regions of human DNA? Means it is asking, just in other words, that is, which of the method you can't use if you want to study a single locus, if you want to study a single nucleotide, right? So in VNTR regions, if you want to study single locus probe analysis, which of the method you cannot use? See, DNA extraction essentially you'll have to do, because that is the only procedure by which you can get DNA in your hand, or you can get your sample, right? So if it is talking about human DNA, then DNA extraction is essentially important in order to study it. So the option cannot be correct. It is asking about not. So the B is restriction endonuclease digestion. We all know that restriction endonucleases are specific, so they will cut wherever we want. So the thing is,